All right, so now we're ready to start on exercise 2, which is gram staining. In this exercise, you will collect a colony from each half of the agar plate used in exercise 1 and gram stain the microbes to identify cell wall structure. Refer to the laboratory bacterial morphology and staining techniques to recall the gram stain theory. So the first step of the procedure is to clear your work area and gather all materials for the listed experiment. So my workspace is clear and I have the materials off to the side. Step 2, wash your hands thoroughly with soap and warm water. Alright, so I washed my hands. Now we're at step 3. Put on safety gloves, face mask, apron, and goggles. Alright, so I got my gloves, face mask, apron, and goggles on. So now we're at step 4. Disinfect the work surface by wiping the work surface with a 10% bleach solution. So it looks like I'm going to need to make some more of the 10% bleach for my next lab, but I have enough to do this lab. Down and let this air dry. Now we're at step 5. Label one disposable cup DW for distilled water and another cup alcohol before adding either the distilled water or alcohol to the appropriate labeled cup. Alright, so I'll just label this one distilled water, and then I'll fill that with some distilled water. And then I'll label this one alcohol. And then I'll fill that with my alcohol. So now we're at step 6. Label the end of a blank slide pre with a permanent marker and then flip this slide over and draw a dime-sized circle on the underside of the slide. So I'm just gonna do it on this piece of paper so you can actually see what I'm doing. So just grab a slide and then we will write pre on top, and then we'll flip it over and draw a circle about the size of a dime, and then flip it back over. And then step 7 is sterilize the inoculation loop by placing it in a cup of alcohol for 30 seconds. So you'll take your inoculation loop and just stick it in your alcohol, and I'll give it a minute just to be extra careful. And then it says remove the loop and shake briskly to dry. And like I said in previous videos, if you're shaking it around, it will basically attract anything in the air to stick to the liquid. And so you'll also be drying it faster, but you're more likely to get contamination. So what I have been doing is I take it out of the alcohol and then just tap it to get any large drops off of the loop, and then I just let it dry for, it seems to be about 2 minutes for it to get most of the liquid off. Alright, so now I will tap off the large drops and let this air dry. And so I'll be back in 2 minutes and then we can get started on step 8. Alright, so now it's been 2 minutes and we can move on to step 8. And let's look at it. It should be mostly dry. There's just a couple little tiny, it's not quite picking it up, but it's mostly dry. There's just little tiny bits that are still on the loop. So now we're at step 8. Submerge the sterilized inoculation loop in the cup of distilled water and then transfer the water adhering to the loop to the dime-sized circle labeled on your slide. And then it says see figure 8. So that's figure 8. So we'll submerge it into our distilled water. And then when you take it out of the water, there'll still be, well, that's interesting. Normally water will stick to the loop, and I'm not seeing it stick in the loop. So maybe going in this way, you'll get more water. So if you look at it from an angle, it did transfer a little bit of water. And then we're at step 9. Sterilize the inoculation loop a second time and then shake to dry. So we'll stick it back in the alcohol and we will wait. And then we'll wait 30 seconds for it to sterilize the loop. All right, it's been 30 seconds. So now I'll just tap off any large drops of alcohol and let this air dry. So I'll be back in approximately 2 minutes. All right, so now we're at step 10. It says collect an isolated colony from the pre-section of the agar plate by touching the sterile inoculation loop to the microbes. So you probably can't really see, but I'm going to try to collect one of these really small colonies in this area. And so you'll want the agar side up. So you'll want the agar side up and you'll grab your colony from the bottom with your loop. And then you'll transfer it to the liquid on your plate. And just mix it into the area that you marked, the circle. And then step 11, immediately transfer the colony from the loop to the area of the slide containing the distilled water. So you can see, yeah, there you can see it. Step 12, stir the material adhering to the loop to the drop of water until dissolved. So you might not be able to see that. It transferred a really small amount of water. Let's see. So yeah, you can kind of see it. But I just tried to stay within that circle and did it a bunch of times so that it definitely would mix. It says, note, failure to diffuse the sample microbes into the water drop will produce inconclusive staining results. So I think what they mean is that if it's like thicker in some areas and not in others, that you might see different results with the gram stain. So just try to make sure it's about the same concentration throughout that dime-sized circle. So now we're at step 13. Place the inoculation loop into undiluted bleach prior to disposal. So I'm going to grab, so I'll just put this here. I'm gonna grab a 1/3 cup and I'll label it bleach, and then I'll put undiluted bleach in there for my waste. So I'll stick this in my bleach. And then step 14, allow the slide to completely air dry. And then step 15, repeat step 6 through 14 for the post half of the auger plate. So we'll grab a second slide and then we will do the same thing, but we'll call this one post. You turn it over and draw a dime-sized circle. And so you'll grab a second inoculation loop and put that in your alcohol for 30 seconds. All right, it's been 30 seconds, so now we'll just tap off any large drops of alcohol and let this air dry for 2 minutes. All right, so it's been 2 minutes, so now we will collect, so we'll submerge it in the distilled water and we will transfer a loop full of distilled water to the dime-sized spot on the post slide. So you can kind of see it. So it just doesn't, it kind of like beads up on the glass. And so then we will stick our loop back in the alcohol and wait 30 seconds. All right, it's been 30 seconds, so now I will tap off the excess alcohol and let this air dry for approximately 2 minutes. All right, it's been about 2 minutes, so now we're at step 10 again, but we're going, instead of collecting a colony from the pre-section of the auger plate, we'll collect a colony from the post-section of the auger plate. And I'm going to probably take one of these colonies because there's probably about 5 of those, no, there's 4 of that type growing on the post side of the plate. And they're big enough that I'll definitely be able to grab them. And then you'll just mix it into the water. So in a circular motion, just keep mixing it into that dime-sized area. And so we'll look at it and yeah, you can see it right there at that angle. There's quite a bit more on this one, mostly just because that colony is so much bigger than the one I took from for the pre side. And so now we'll put our inoculation loop into the bleach. And so step 14 is allow the slide to completely air dry. And so I'll give an extra 5 minutes just to make sure that both of these are fully dry before moving on to the next step. But the pre-slide will be drier than the post-slide since we did that first. So you can start on the next part as soon as you can tell that it's fully dry. You can barely see, but there's like a thin layer within that dime-sized, and it'll look matte when it's fully dry. If it looks shiny in any of the areas, then it's still wet and you'll need it to dry a little bit longer. And then it says, note, collect a colony with a different morphology than the one used for the pre-slide if possible. And so I kind of wish they would put these, a lot of times it'll tell you like after you've done it. Fortunately, I did do that. In the pre, area, I took like the really, really tiny colonies, which are also growing on the post side, but I chose the bigger colonies on the post side. So that would be something to keep in mind. Step 16, light the candle with a match or a lighter and then heat fix the pre-sample by holding the slide with a test tube clamp while passing above the flame 5 times. And then it says see figure 9. So that's figure 9.